Assalamu alaikum students. I am Dr. Erum, Professor and Head of Department Physiology at FRPMC. My email address is drerumimran@gmail.com. Today is the second lecture of Physiology in the Neurosciences, that is the Physiological Anatomy of Synapse. Now, what is okay, now let's see the learning objectives of this lecture. By the end of the lecture, you should be able to define what is synapse, define neural synapses and discuss its types, explain the physiological anatomy of chemical synapse, explain why synapses operate only in one direction from presynaptic to postsynaptic neuron, compare the events at excitatory and inhibitory synapses, discuss the properties of synaptic transmission, and discuss the factors affecting synaptic transmission. So, what is a synapse? Actually, synapse is a junction between two excitable cells. As you all know that we have got two types of excitable cells in the body, that is the neuron and the muscle. So, the synapse is a functional junction. It's not the anatomical junction, it's the functional junction between the two excitable cells. Now, it may be between two neurons, it may be between a neuron and a muscle, which you have studied in a neuromuscular junction, and it may be between two muscle cells like two cardiac cells connected via gap junctions. So, it may be between two neurons, between a neuron and a muscle cell, and between two muscle cells. So, it's a site where the information is transferred from one excitable cell to another excitable cell. This is one neuron, you can see, and this is another neuron, and there is a functional communication between the two neurons, which is known as the synapse. So, there is no anatomical continuity. You can see that they are quite separate. So, there is no continuity between the two cells involved in the formation of synapse, but it's a functional junction between the two excitable cells. And this typical diagram showing the example of a synapse between two neurons. Now, we will focus, since it's a neuroscience, so we will not discuss the neuromuscular junction. We have already done that. We have also done the cardiac muscle, in which two muscles they are connected via gap junction. We will focus here on the neural synapses, means the synapse between the neurons. So, these synapses are between the two neurons. Then those synapses are known as the neural synapses. Now, information is actually transferred in the nervous system in the form of action potential, as we know that this action potential then travels from one neuron, then it reaches the axon terminal, and then it excites the other neuron. So, how the impulse transferred from one neuron to another through a junction which is known as the synapse, and this is how the information reaches through a succession of neurons, one after another. Now, neural synapses, they are of two types. They may be a chemical synapse between the two neurons, or they may be the type of electrical synapse between the two neurons. Now, the most important is the chemical synapse. Less important in the neural synapses are the electrical synapse. These electrical synapses, they are commonly found in smooth muscles and in the cardiac muscles. In the nervous system, there are very few electrical synapses, and they are not yet so much discovered, but they are few. But most common synapses which are present in the neural synapses, they are of the chemical synapse type. Now, electrical synapse, as we have discussed before, that these are nothing but they are the ion channels, and they conduct the electrical impulses from one cell to another. They have connected via gap junctions, and it's a two-way type of conduction, and it's less time consuming because the ions, they move directly from one cell to another cell through the gap junctions. They are mostly found, as I told you, in the smooth muscle and cardiac muscle. In the CNS, they are rare to find, find these type of electrical synapses, but there are some circuits which are responsible for sleep, mental attention, emotions and memory. They do have electrical synapses in which the electrical signals from one neuron may pass to the another neuron, but not much is known about the electrical synapses in the neurons. The most important synapses present in the neurons is the chemical synapses. So most of the synapses used for the signal transmission in the nervous system, they are the chemical type of synapses in which a certain chemical is released. Since it is released at the nerve terminals, so this is known as a neurotransmitter. These are the two neurons. You can see this is neuron 1, this is another neuron. So these two neurons, they are connected via chemical synapse in which a neurotransmitter is released by one neuron. So a neuron which is conducting the impulse towards the synapse is known as a presynaptic neuron. So this is a presynaptic neuron. This junction between the two neurons is known as the synapse, and the neuron which conduct the impulse away from the synapse is known as the postsynaptic neuron. Now you must understand the presynaptic word and the postsynaptic. Before synapse, the neuron is known as the presynaptic neuron. This neuron is capable of releasing the neurotransmitter in the synapse. Okay, so impulse is carried towards the synapse by this neuron, which is known as the presynaptic neuron. Now the impulse which the neuron which conduct the impulse away from the synapse is known as the postsynaptic neuron. So again you can see in this simplified diagram, this is a presynaptic neuron which is carrying the impulse towards the synapse, and this is the postsynaptic neuron which is carrying the impulse away from the synapse. And here in the synapse, a neurotransmitter is released by the presynaptic neuron. Now let's discuss the functional anatomy of the chemical synapse. Keep in mind the functional anatomy of the neuromuscular junction is much as similar as to that of the synapse between the two neurons. It's quite similar to the synapse between the nerve and the muscle membrane. Okay, but here there are two neurons. This is a presynaptic neuron, this upper one, and this is the postsynaptic neuron. There are two neurons and they are connected via synapse, and this is a synapse. So now the neurotransmitter is synthesized in the presynaptic neuron. This is a presynaptic neuron. This is postsynaptic neuron. So here you can see different vesicles, they are present in which a neurotransmitter is being synthesized and stored over there. Okay, so what happens when an action potential arrives at the axon terminal? You can see the action potential is arising. So what is happening, that neurotransmitters, they are formed in these vesicles and they are stored over there. Okay, so what happens when action potential reaches the axon terminal? Voltage gated calcium channels open at the terminal and calcium move inside, and which causes the membrane vesicles to fuse with the, now these vesicles are fusing with the plasma membrane and they release their neurotransmitter in the synaptic cleft. Now this neurotransmitter then binds with the receptors which are present in the postsynaptic membrane. So the neurotransmitter is present in the presynaptic terminal and the receptors, they are present in the postsynaptic membrane. The released neurotransmitter molecule then bind with the receptors which then open up the ion channels. You can see that the ion channels here are closed, but as the neurotransmitter come in bind, these ion channels, they open and the ions, they move inside or outside the postsynaptic membrane. So once its action is completed, this neurotransmitter is now ready to be deactivated either by the enzymatic degradation. If you remember, in case of acetylcholine, there was an enzyme acetylcholinesterase which break this acetylcholine and the action then stopped. So this is how the presynaptic neuron activates the postsynaptic neuron by the release of a neurotransmitter, and this neurotransmitter action is finished by the either the uptake of the neurotransmitter back into the presynaptic terminal, or it is degraded by the enzymes. So it depends on the type of neurotransmitter. So, importance of chemical synapses in the nervous system. Now, as we have seen that this type of neural synapse, the chemical synapse, act as unidirectional belts. They only allow the flow of impulses from pre to post synaptic neurons, because the neurotransmitter is released from the presynaptic terminal and the receptors, they are present in the postsynaptic. So, always the flow of impulse will be from presynaptic towards the postsynaptic. Okay, so this phenomena is known as principle of one-way conduction. There is a one-way conduction. It is very important. It means the impulse is going in only forward direction, or we say that orthodromic direction, so that the specific goal will be achieved. Otherwise, what will happen? The impulse will move in both direction, and the impulse will be lost. So to perform a desired function of different functions like sensations, motor control, memory, any function, the impulse must travel in a forward or the orthodromic direction from presynaptic towards the postsynaptic. So these synapses, they are also the sites in the nervous pathway at which transmission of impulse can be accelerated, slowed down, or blocked. So they are the site for the impulse, where the impulses can be accelerated, they can be slowed down or blocked either by physiological, pathological, or pharmacological influences. You will study that few drugs, they act on these receptors, and they may either increase the activity of these synapses, or they may decrease the activity of these neurotransmitters. So they are the important site where the impulses can be accelerated, slowed down, or blocked. Okay, now let's discuss the type of receptors which are present in the postsynaptic membrane. As I have told you that the membrane of the postsynaptic neuron has a receptors on which the neurotransmitter comes and binds. So it consists of large number of receptors, and the receptor may be of two types. Either they may be ionotropic receptors or the metabotropic receptors. Ionotropic or metabotropic. Now both of these receptors have two components. Whether it is ionotropic type of receptor or metabotropic type of receptor, they have two similarities. What are those similarities? That they have two components. Here you can see it's a binding component, the binding component which binds the neurotransmitter. The neurotransmitter comes and binds over here. And second component is the ionophore component. This ionophore component which actually passes all the way through the cell membrane. So each receptor, whether it's ionotropic receptor or metabotropic receptor, it has got two components. A binding component. Here another example, you can see it's a binding of the neurotransmitter binds to a component which is known as the binding component, and it binds a neurotransmitter like a lock and key mechanism. Okay, and second component is the ionophore component. This other component is whole receptor, and this ionophore is this part which spans throughout the cell membrane. So this is the example of the ionotropic receptor, and this is the example of the metabotropic receptor. Now let's discuss the first ionotropic type of receptor. The ionotropic type of receptor, again I told you, whether it's a metabotropic or ionotropic, it has got two components. The binding component, you can see here that how the neurotransmitter fits onto the binding site like a lock and key, and neurotransmitter, it binds, and they receptors open the ion channels. Binding of the neurotransmitter open the ion channel. This ion channel gets open. That's why these receptors, they are known as ionotropic receptor, because they open an ion channel. Okay, now these ion channels, they can be a cationic channel or it can be an anionic channel. The cationic channel, which allows the positive ions to move, like sodium ions, potassium ions and calcium ions. Okay, so whenever the neurotransmitter comes and bind to the binding component of the ionotropic receptor, the ion channels, they open up and there is a tremendous movement of ions inside and outside. These channels can be a cationic channel. Here the example is shown of the cationic channel in which sodium is moving from outside. As you all know that more sodium is present outside, so it will move inside. The ion channels, they are responsible for the passive movement of the ions, okay, for diffusion. It allows the diffusion. So sodium is going to diffuse from high outside concentration towards the lower inside, okay. So it is moving down the concentration gradient into the cell. So neurotransmitter substances that open the those neurotransmitter substances that bring the positivity inside, due to the opening of the channels, the positivity when come inside, it excites the membrane towards the threshold value and the membrane gets excited. So such type of neurotransmitter that open the sodium channel, sodium channel is known as the excitatory neurotransmitter, because when sodium moves inside, it will cause excitation. So the examples are acetylcholine, norepinephrine, glutamate, to name few. There are also others neurotransmitter which opens the cationic channels for the sodium, and sodium influx lead to the excitation of the neurons. There are other neurotransmitters which can open the anionic channel. Okay, so the anionic channel when open, what will happen, that chloride ions will pass through. Okay, the anionic channel, the negative charge ions, for example, the neurotransmitter comes and bind, the ion channel open and the chloride ion will move from outside to inside. Again, the gradient of the chloride is from outside. There is more chloride in the ECF as compared to ICF. So chloride will move from outside to inside. Since chloride is a negative ion, so when chloride moves inside, it will create more negativity and the threshold will be far away and the membrane potential will be not excited and the membrane will not be excited, and we say that such a neurotransmitter is the inhibitory neurotransmitter because it moves the membrane potential far away from the threshold for excitation. So the neurotransmitters which open the chloride channel, they are known as the inhibitory neurotransmitters, and to name is glycine and GABA. They act as the inhibitory neurotransmitter. Sometimes dopamine acts as excitatory, and at some places in the nervous system, it acts as the inhibitory neurotransmitter. So what will happen if a neurotransmitter binds and open the potassium and calcium channels? Let's see. They are also the cationic channels, but what happens if the potassium channels are open? If a potassium channel is open, since more potassium is present inside, so potassium will leak outside. It will move outside from inside, creating more negativity, and the membrane will reach far away from the threshold for excitation. So any neurotransmitter which open the potassium channels will also cause inhibition. Okay, and opening of calcium channels, if a neurotransmitter binds and open the calcium channels, the calcium will move inside, and again the calcium is a positive ion coming inside, creating positivity inside, and membrane potential may reach to the threshold value and may cause action potential. So opening of sodium and calcium channels will lead to excitation, and opening of potassium and chloride channel will lead to inhibition. So depending the type of neurotransmitter, we can say that if it is an excitatory neurotransmitter and it opens the sodium and calcium channel, it will cause excitation. If a neurotransmitter opens the potassium and chloride channels, it will cause inhibition. So another type of receptors, they are the metabotropic receptors. Again, in this type of receptor, I told you also there are two components, the binding component and the ionophore component. The binding component over here, you can see the neurotransmitter comes and bind to the receptor protein, and this is actually an ionophore component, and this is linked to a protein which is known as G protein. So it's G protein coupled receptor. So once the neurotransmitter come and bind, and then this G protein gets activated, which has got three subunits, alpha, beta and gamma subunits. Once it gets activated, the alpha subunit is detached from this complex, and this alpha subunit, now it performs number of actions. You can see now it can open the ion channels, or either it activates some enzyme, membrane enzyme like adenyl cyclase and guanylate cyclase, which converts the ATP and GTP into cyclic AMP and cyclic GMP, or it activates the process of gene transcription and results in new protein formation and produce the structural changes, or it activates one or more other intracellular enzymes, and then those enzymes then produce the desired response. So these are the series of reactions which are occurring in a metabotropic type of receptors. Now let's summarize. The receptors have two components, the binding site or the binding component that bind the neurotransmitter, and ionophore component which passes all the way through the membrane. So there are two type of receptors, either they may be ionotropic receptors or the metabotropic type of receptors. The ionotropic receptor, they open the ion channels, which may be the cationic channels or the anionic channels. Okay, the cationic channels may open if a neurotransmitter binds and open the sodium, potassium or calcium. Okay, so these are three positive ions. So opening of sodium channels increase the membrane potential towards the positive direction, means the threshold level is reached and excitation of the neuron is going to occur. Similarly, opening of the chloride channel will lead to the negative charges movement inside the neuron, and membrane potential will become more negative and away from threshold, so leads to inhibition, okay, not the excitation. In a metabotropic type of receptor, it's a second messenger system, and this is very important where we need the prolonged post-synaptic changes are needed. For example, in the memory. Okay, in the memory, the structural changes, you will study memory in detail, in which you will see that the structural changes are required, and these structural changes, they are produced by these metabotropic type of receptors will produce the formation of new proteins and structural changes in the neuron, and they actually help in the formation of the new memory tracts. So now we have seen that some neurotransmitters, they excite, whereas others inhibit the postsynaptic neurons. So each presynaptic neuron release only one type of neurotransmitter. On binding with receptors, different neurotransmitters will cause different ionic permeability changes. Some will open the sodium channels, some will open the potassium channels, and on the basis whether they are doing excitation or inhibition, they are named as excitatory or inhibitory neurotransmitters. Okay, so how excitation is brought about by the presynaptic neuron? If a neurotransmitter is released from the presynaptic neuron and it opens the sodium channels on the postsynaptic neuron, so it will cause excitation, more sodium influx leading to excitation, or excitation can occur if there is a depressed conductance through the chloride and potassium channels. Okay, or there may be increased number of excitatory receptors or decreased number of inhibitory receptors. All these four factors will lead to the excitation of the postsynaptic neuron. Now, how the inhibition occur in the postsynaptic neuron? If a neurotransmitter is released from the presynaptic terminal and it opens the chloride channel, what will happen? The chloride influx will occur, causes more negativity. So such a neurotransmitter is the inhibitory neurotransmitter because it is opening the chloride channels. Moreover, if there is increased conductance of potassium, more potassium will leak out, membrane will move away from the threshold and leads to inhibition. increased number of inhibitory receptors or decreased number of excitatory receptors. All these four factors will causes inhibition in the postsynaptic neuron. Okay, now we will discuss the properties of synaptic transmission. This is very important for your SAQs and SAQ question of the neuroscience portion, that you get a small essay type question on the properties of the synaptic transmission. So, the first property is one-way conduction. As I told you before, that synapses causes the conduction of impulse in one direction from presynaptic to postsynaptic. We are talking about the type of chemical synapses in which a neurotransmitter is released from the presynaptic terminal, which binds onto the receptors which are present onto the postsynaptic membrane. So, there is always a one-way conduction from presynaptic towards the postsynaptic, and this is known as the Bell-Megendri law. Okay, so it's named after the scientist who discovered this, and I told you that this one-way transmission is very important to move the signals or to move the impulse in a desired goal. Now, second is the Dale's principle. Second property of the synaptic transmission is the Dale's principle, and it states that a given neuron contain and release only one neurotransmitter and exert the same functional effect at all of synapses. So, only one type of neurotransmitter is released from one neuron. It is not so that one neuron at one site it is releasing acetylcholine and other site it is releasing norepinephrine, other type. It is not so. Only it has a factory to synthesize only one type of neurotransmitter. So a neuron only release and synthesize only one type of neurotransmitter at all of its nerve terminals. Okay. So naturally it is going to produce the same effect at all of its synapses. For example, if a single neuron is making about 3 to 5 terminals, so at all the terminals it will release only one type of neurotransmitter and it will produce the same effects at all of its synapses. This principle is known as Dale's principle. Third property of the synaptic transmission is the synaptic delay. Now there is a delay of about 0.3 to 0.5 milliseconds during the transmission of a signal from a presynaptic to a postsynaptic neuron. How much delay? About 0.3 to 0.5 milliseconds of delay. Now, from the delay time, what we can calculate? We can calculate the number of synapses in the pathway. Okay. For example, if there are about, if the delay time is about 0.5 milliseconds. So, what we can say that it's only made up of one synapse. If it's double, for example, 1 millisecond, then we can say that there are two synapses. If more than one, we can say they are polysynaptic. Okay, so it can tell us about the number of synapses in the pathway. By the calculation of this delay time, we can calculate the number of synapses in the pathway, whether it's monosynaptic or polysynaptic. Monosynaptic means one synapse and poly means more than two synapse. Disynaptic means two synapse. Okay, why there is a delay in the transmission of nerve signal from a presynaptic to the postsynaptic. There are various factors. First of all, discharge of the transmitter substance by the presynaptic terminal. As you know, there's some delay occurs due to the release of the neurotransmitter by the presynaptic terminal, because the action potential when arises at the nerve terminal, the voltage gated calcium channels open and the transmitter vesicles, they fuse with the membrane and then they release the transmitter substance into the synaptic cleft. Then diffusion of transmitter substance to the postsynaptic membrane. Once it's released, it slowly diffuse and move towards the postsynaptic membrane. Then it binds with the receptors on the postsynaptic membrane. Binding will take time, but after binding, it will alter the membrane permeability is going to be changed. Then what happened that sodium ions will move to the inside or outside. Okay, so ionic permeability will be altered and ions will move inside and outside and cause excitation or inhibition. And finally, in the postsynaptic membrane, summation of postsynaptic potentials, they will be summated. So all these factors, they contribute to the synaptic delay, and by this synaptic delay we can estimate the number of synapses present in the pathway, and the normal delay is about 0.3 to 0.5 milliseconds. Fourth property of the synaptic transmission is the fatigue of synaptic transmission. Now what happened is, fatigue is actually a decline in response of postsynaptic neuron after long period of frequency stimulation of the synapse. If a synapse remain active for a prolonged time, what will happen? It is going to fatigue. So it is manifested by a prolonged synaptic delay, then failure to transmit some or all of the impulses across the synapse. A time will come when there will be no impulse transmission across the synapse. It means the synapse has fatigued. So it's because what is the mechanism of fatigue? Because of the exhaustion of the stores of neurotransmitters. Now due to continuous stimulation, continuous activation, what happens? All the stores of the neurotransmitters, they are exhausted, and now the neurotransmitter is not available for the synaptic transmission. So we say that our synapse has got fatigue. So there may be other reason. The main reason is the exhaustion of the stores of the neurotransmitter. Also, inactivation of the postsynaptic receptors due to the accumulation of metabolites. Okay, the different metabolites, they get accumulated, ionic concentration changes at the synapse terminal, which lead, and third is actually the abnormal concentration of ions. So this all contribute to the fatigue of the synaptic transmission, but it's a temporary phenomena. What happens once the neurons, they pass in the stage of resting stage, the resynthesis of neurotransmitter occurs and the impulse conduction across the synapse is then recovered. So it is very important, the fatigue of synaptic transmission. It's a protective phenomena, and it is very much helpful in a person of epilepsy, because in epilepsy the neurons get overexcited, and due to overexcitation there is a fatigue of the neurons, and fatigue of the neurons will lead to the halting of the actions of the neuron. Otherwise the person will remain in the state of fits. Okay, so now let's discuss another property of the synaptic transmission, which is the convergence and divergence. Now here in the first diagram, the convergence is shown, in which multiple presynaptic neurons, they are ending on a common postsynaptic neuron. Means many neurons, they are converging, okay, towards a common postsynaptic neuron, and this property is known as the convergence. And other is the divergence, in which what you can see that impulse from one presynaptic nerve fiber is ending on multiple or a large region of postsynaptic neurons, large number of postsynaptic neurons. So this phenomena is known as divergence. The impulse is diverged from one neuron to multiple postsynaptic neurons. So both of these type of transmissions you will see are present in different pathways. So in some pathway you will find the phenomena of convergence, and in some pathways you will find the phenomena of divergence. Another property is known as the summation. Okay, so whenever the stimulus of subthreshold strength is applied, there will be no development of action potential in the postsynaptic region. At least we need a threshold stimulus so that a threshold potential is achieved and action potential will be generated. But what happens if multiple subthreshold stimuli are applied? These effects will be summated or added up together, and then they can lead to the development of action potential in the postsynaptic membrane, and this property is known as summation. Now let's discuss the type of summation. There are two types of summation, the spatial summation and the temporal summation. So this is the temporal summation. In the temporal summation, what is happening over here? Look at this. These are the three presynaptic neurons, and this is one postsynaptic neuron. Now what is happening in the temporal type of summation? presynaptic neuron stimulated will be the same. Okay, but many stimuli